Okay, we'll get started. Hi everyone, good afternoon and thanks for joining today. This is the inaugural Professor Yanis Yanas lecture. It's a real pleasure to be here and to see you all. I won't talk for long, but just to say Professor Yanis Yanas was a pioneer of regenerative medicine and the field of tissue and organ regeneration. And he invented artificial skin for the treatment of severe burns. And Yanni was a beloved member of the Department of Mechanical Engineering here for almost 60 years as a colleague and friend. So it's a real honor today to have this inaugural lecture, Professor Yanis Yanas lecture in bioengineering. And with that, I'm going to hand you over to Professor Myron Spector. Myron was a colleague and collaborator and co-instructor with Yanni for many years, and he's going to introduce our inaugural speaker.
Thanks. Nice to see you all, and you're right to be smiling. Yanni brings smiles. He does bring smiles. There's really fond memories, and he did so much for so many people, including us at helping. He helped us in many ways, not the least of which is by being a role model. And we are always playing roles. We have to play a role. We're asked to do something, and so many times, I don't know about you, but I wonder, what am I supposed to do? Well, in many cases, I said, well, what would Yanni do in this situation? Most of the time it worked. Not always, but most of the time. But I want to continue that because there's no better speaker today as the inaugural address than Brendan Harley. Because if I was approached by a young person coming into the Institute and they wondered, what should they be doing? How are they going to satisfy different roles expected? I'd probably say, just go back and see what Brendan did. Mm-hmm. Okay, and it's in so many ways. Now, Brendan got his bachelor degree in the year 2000 from Harvard. Okay. Okay. But then he saw the light. He came to the Institute for his master's degree. He got that in 2002. And then he obtained his Doctor of Science degree in mechanical engineering in 2006. That means this is his 20th anniversary. So there's no better person to have as someone coming back to celebrate the 20th anniversary of his or her doctoral work. But let's continue with a role model. Let's continue a role model with Brendan. You know, Brendan took advantage of all the accessible resources at the Institute. And this means not just labs, but also people, faculty. So for example, Lorna was his principal thesis supervisor. Yanni was his co-supervisor. Then there was Peter So, Paul Mazzadera, who were on his thesis committee. Merton Flemings. It's been so long since I used that name. Then from outside the country, Fergal O'Brien, right? Fergal O'Brien from Royal College of Surgeons in Ireland, and also a fellow named Andrew Lynn, bright kid, University of Cambridge. So I think Brendan followed where it would take him to meet people who could teach him and who could serve as his role models for it. You can see this. You can see how all this played out with the role models in the journal publications. The number of people, co-authors, including myself. I had a couple. That as well. Now, a second point, because I'm learning lessons from him. He's already served in my role model. I wonder what the hell is he doing with glioblastoma? Oh God, that's so frightening. Well, here Brendan took advantage of opportunities to get into some new investigative areas. And he followed that. And he's been following that. And he told me, and I checked it, fact-checked you. Yep. Just on Tuesday, the National Cancer Institute of NIH announced that the Cancer Center at Illinois will be the 8th National Cancer Center. So it's a big deal. It hasn't happened in a long time since the 7th one was appointed. And there are 2 research programs. The one in cancer engineering and biological systems is co-led by Brendan. So this is another way that he's extended what his capabilities are to get into other important areas. And finally, his collaborative nature just in general, in engaging others, as I mentioned, reflected in the co-authorship. So I could go on, Brendan, but I want to hear what you have to say.
Well, thank you, Myron. All right. Hi, everybody. So I'm Brendan. All right. This is a different lecture to give. I give lectures quite often. It's something special to come back and talk about someone who meant a lot to you. So I'm going to talk today about regenerative biology, which I think Yanni would have really enjoyed, and engineered tissue models, which he might look at me weird about. Why are you not putting stuff into patients? Why are you trying to do this thing in the laboratory in a different way? But I think he would understand. And so I'm going to tell some stories about how my lab has developed over the last 20 years, where we started from, and some of the stuff I'm excited about now. But first I want to give a nod to Yanni. I think the reason that the field of tissue engineering is where it is at is some of the work that he started years ago, 50 years ago, starting to play around this idea of how do we treat burn patients better? How do we develop systems that might actually lead to healing? And that was not the goal initially. The goal was to try to develop an improved bandage, a way to temporarily splint large-scale burn patients. But over the years, they developed this porous collagen material, a collagen glycosaminoglycan scaffold, went through process of optimizing it in preclinical work, in clinical trials. It's been a transformative thing to grow back, as Yanni says, skin, not scar, right? That was the tagline attached to this, is how do we think about fundamentally changing how tissue heals and opening up an avenue of what we might do into the future, right? And I think it's and the number of years that I know he fought through NIH not thinking this was even a thing, and in a process where that he had to really prove it over decades that this was a clinically relevant solution that has now touched hundreds of thousands, if not millions of people. And I think that's a really amazing legacy to live. But as you mentioned, Yanni is also a mentor. He's someone who has meant a lot to me personally. I am a generation of the ear mouse. I was in high school when the ear mouse was on the cover of the Boston Globe, and I was hooked. I thought biology and engineering is where I wanted to be. And in fact, this is a quote. Every now and then you write garbage. These two sentences up there are something I had in my graduate application in the late 90s when I applied to MIT. One of my interests is in the area of tissue and organ growth. While predominantly biological, these processes are mechanically oriented and may be addressed using engineering analysis, which I actually think is pretty prescient in terms of where we ended up being. So I wrote that. And 3 months later, I got an email from Yanni. And this is just part of it, but those people I met with today, I saved the text that I still use to this day when I want to write to a graduate student I want to recruit, right? And he wrote a long email to me that, among other things, he was also a Harvard graduate. I'm wearing my Dunster tie because he was an Adams House graduate. I never wear this tie, but I'm wearing that today for that. But he said, I'd like to offer you a research assistantship. And there was a much longer thing about it, but what he said is understanding the molecular biological mechanism of regeneration in nerve is at a particular premium, since understanding should make this regenerative approach applicable to other organs. He wanted me to join the lab to work in an applied tissue engineering project. He wanted me to think about identifying underlying mechanisms of how this works so that we could scale it out bigger and think about how do we use this knowledge as a foundation for a future of growth. He also mentioned on the side, it looks like my lab might be involved in a future collaboration with the University of Cambridge in the area of the biology of extracellular matrices, right? So I met with him and we just sat in his office where he had all of his papers on the wall that we could look at, and sat on the couch and talked through projects, and I agreed to join his lab. And so my process of being a graduate student started with discovery. My first project was very much an applied tissue engineering project. We were interested in how do we engineer cylindrical conduits that could be used for peripheral nerve regeneration. So we're doing sciatic nerve grafts across the back of rats and looking at the rate of degradation of these tubes to try to understand how the tube degraded away. Did that affect the quality of healing? We developed some metrics by which we could assess successful regeneration across animal models. One of the big challenges one have in looking at healing is how do you compare between what one lab did and another one lab did. So we developed some metrics by which we could look at qualitative processes of healing in a now quantitative way. That was the discovery phase of my thesis. As mentioned, I then was jointly advised by Lorna and Yanni, and we thought about how do we develop better mechanistic understanding of how these biomaterials work. How do we take what was green cells living in a red scaffold as we might be able to image it 20 years ago? How do we develop mathematical approaches to describe the structure of these materials? So we adapted some cellular solids methods to describe its mechanical properties. We then use this to understand things such as cell invasion and contraction, which are fundamental to how healing occurred in our nerve models. The goal is using this to try to develop a new generation of tissue engineering products. And then we did get to work with the University of Cambridge and Andrew Lynn and Bill Bonfield and the group over in the UK, where we developed one of the first biphasic biomaterials that we've used for osteochondral repair in the knee. Got all the way into clinical trials and started a company. And so what Yanni said in that one email he wrote me, we're going to do applied work, we're going to develop mechanism, and then we're going to scale it out to something bigger. That turned out to be my entire PhD. And it gave me a model by which we could think about developing enabling tools and thinking about broader applications. And it meant we worked collaboratively, with Peter, and you forgot Doug Loffenberger and Mohammed Zaman before he became faculty. And so he was a model of finding people and bringing them together. And it's been something I've tried to do for the rest of my career. And so, as I told a few people today, I thought about how I might want to do this and what Yanni would like to see. So I thought I'd share some of the work we're doing, why we're doing it, what is the new stuff we're excited about, and how did it stem out of this observation of applied mechanisms scaled back to think broader. And I think that's a really, it's a microcosm of, I think, what academia needs and what is really beautiful about academic science. And so it's been an inspiration. So what is it that we do? So my lab is, we are inspired still by this idea of how biology and mechanics come together. We're just inspired by the tissue environment that inhabits all the cells and tissues of our body. Right? So we think about most tissues in our body, they're not uniform sacks of amorphous cells living in an amorphous structure. They have structure. We have cellular heterogeneity. You have stem cells and mature cells and vascular cells and immune cells that are working together collectively. These tissues are hierarchical or multi-scale. They have structure attached to them, right? They have spatial transitions if you think about musculoskeletal interfaces, and they change in time. As we development, through disease, through aging, through regeneration, we wanted to start thinking about developing tools to gain access to this idea of the heterogeneity innate in our tissues. Back when I was a graduate student, we spent a lot of time trying to make uniform materials so we can gain mechanistic insight. The next challenge is, how do you scale it out to start developing patterns? And I would argue, going into the future, there's a new challenge that we face. And so we can start thinking about the next challenge for biomaterials, and how do we move beyond generalization? As engineers, we think a lot about solving problems under constraints and coming up with an optimal solution. And the reality is, if I tried to develop a bone tissue engineering scaffold, which I'll talk about in a minute, and use it on all of you, it would work differently in all of us, right? As a function of age, as a function of underlying disease, as a function of sex, potentially. Lots of things may affect how healing happens. And so it's important for us to think about, how do we develop biomaterial systems that account for this heterogeneity? I'm not talking we need 7 billion new solutions, but there's a need to try to understand not just a solution for bone tissue engineering is done and dusted. We want to think contextually. So I'm going to talk today about a couple different vignettes of what we've been up to, and hopefully it's entertaining. One of the big areas that we've been doing, and is what I wanted to start with, is an adaptation of what Yanni had been doing for a very long time with this collagen scaffolds. We were very interested in adapting into new systems. If Yanni was doing skin and nerve, I wanted to move in a different space. And so we started thinking about musculoskeletal applications, but I thought very strong and hard about something Yanni told me from the beginning. Sitting in his office, sitting on the couch as we were talking about different projects, his first question is, will this work for real? Will this get into a clinic? Will this get into a patient? And what are the metrics that you need to get over? Because don't waste your time playing with challenges that won't be clinically applicable, that the FDA will never approve, that will not be surgically practical. How do we make things that have impact and meaning? And so this is the question we've thought a lot in the context of musculoskeletal tissue engineering. I'm going to mostly talk about craniofacial bone regeneration, although we have dabbled in rotator cuff repair and cartilage and osteochondral defects. But really, what does it take? And the argument I'd have is the tissue engineering triad we've used for the last 40 years, which is putting materials, cells, and biologics, is probably broken. It's probably not going to solve all the things that we need to address challenges of heterogeneity, to address challenges of scale, to address challenges of cost. And so that opens up opportunities for new areas. I'm going to talk about some of our work reimagining biomaterials as factories of endogenous factors to affect healing, as ways to alter the immune response, as ways to address surgical practicality. And I'm going to tell it through the lens of the collagen gag scaffold that Yanni became so famous for. So this is an image of the CG scaffold I took in his lab more than 20 years ago. The date is cropped out, but it was one of the early images that I took trying to look at the structure. And so this is a porous collagen glycogen scaffold that is fabricated by taking an aqueous suspension of collagens and proteoglycans. You mix them together, you then freeze it. You get ice crystals that form. They push the collagen and gag components to the spaces between the ice crystals. And if you sublimate out the ice crystals, meaning you drop the pressure and increase the temperature, you turn solid to vapor, and you pull out all the solid. And so Mert Fleming's was a project to try to refine the freezing conditions of making these ice crystals so we can control the size and the shape of the ice crystals, because that would give us control over the size and the shape of these pores. These scaffolds had pore sizes on order of 100 microns, which is about the size of a cell, maybe a little bit larger, so cells can move through them relatively easily. So we're talking about engineering the space that cells live within and providing an environment to control cell activity. And so the question that we started asking from the beginning is, how might alterations to the structure of this scaffold affect cell activity? So at the time, 20 years ago, the rage was putting cells on elastic substrates and measuring how the stiffness of a substrate affected stem cells to differentiate. Our question was, in a porous three-dimensional material, what's the analog? You have a cell sitting on one of these fibers. As I make the pore bigger or make it elongated, the fibers get longer and thicker, meaning they get floppier or less floppy. So changing pore structure actually turned into like a column problem if you're a mechanical engineer, where you have the flexural rigidity of the fiber changes. And so cells could read structure that we produced as mechanics. And we published a whole series of papers early on. We're trying to work out how cells sense the pore structure of these materials, making them highly aligned for tendon applications, making them with different proteoglycan chemistries for immune applications, and making a mineralized version, which has been the forefront of my lab for the last many years, is developing a regenerative implant for bone repair. We focus primarily in the space of craniofacial bone repair. Again, this is an assistant professor hack for those of you on the younger side. When everyone in the field is doing extremity bone regeneration, don't do that. Do something different. We started working in the space of craniofacial bone repair. We have lots of examples of the types of defects you see in the head and neck. These are congenital defects such as cleft palate. You have post-oncologic, so resection after surgeries. You also have traumatic injury, which for most of us would be car accidents or maybe a gunshot wound, right? These are big, they're nasty, they're unique, and they don't heal particularly well. And so the challenge that we're faced is to develop a material that could be shaped precisely and quickly like an alloplastic implant. One of the current standards of care is put in a permanent polymer that will fill shape, but it won't heal, right? And so that works great if you're an adult, maybe. If you're a kid, it's not particularly good if you want to think about healing. The other two approaches is taking a little bit of bone from your hip, an autologous bone graft or an allogenic bone graft from a cadaver. You put that into space. It can heal regeneratively, but surgically it takes a long time to do that surgery. And if you're thinking as a clinician, do I do the alloplastic implant that I'm in and out, or do I spend 4 times as much time doing the regenerative implant? Which one am I going to pick? And so our challenge was to try to make a material that could heal like Yanni imagined, regenerating bone de novo, but could be used quickly and effectively like a piece of plastic that would be sitting on the shelf. And so we thought very much about the structure. We wanted this material had to fit into a unique shape that was different for every patient. It had to affect the immune response that's happening right after injury, and it had to have long-term healing capacity. Yanni would want to see stroma, he wanted to see vasculature, he wanted to see no scar. And that was our basis. So I'm going to show you the punchline before I talk about some of the work beforehand. What we've come up with is a mineralized version of the collagen scaffolds I worked with Yanni. There was a process by which we incorporated calcium and phosphate into a nanocrystalline structure that would embed into the scaffold. So we have a collagen and proteoglycan matrix that is embedded with mineral content, and this works incredibly well. The top line, this is our dermal regeneration template, a collagen gag or CG scaffold. We can take these scaffolds, and this is in a rabbit calvarial defect. This won't heal on its own with our collaborators out at UCLA, where the scaffold on its own, the non-mineralized scaffold, you can see very little healing in the hole. You could add in stem cells, you could add in BMP2. This is the infused product most people use to heal vertebral damage. You see a little bit of bone formation, but mostly soft tissue repair. Whereas this mineralized collagen scaffold, we see really effective infill. We don't need to add stem cells. We don't need to add osteogenic factors. And it was because we spent a lot of time mechanistically developing a material that would aid cell activity. These materials can be grafted, and within about 12 weeks, we grow back more than 60% of the mechanical nature of the bone itself. It continues to heal over a longer period of time, but these are defects that wouldn't heal on their own and are now filled. Thinking about what Yanni would have had me do, well, we did a lot of mechanistic insight. We spent years working on the mineral content of these materials, how the calcium and phosphate is released out of the scaffold, how it activated different signaling cascades, so we get effective osteogenic activity, meaning we could take a stem cell from a patient, we can get them cells to turn into bone cells really effectively. We thought a lot about mechanics. As we start playing with cross-linking induced mechanics of the scaffold, this would activate the mechanotransduction pathways and refining it so that we could get really efficient activity. We played a lot with the proteoglycan content, which I'll get back to in a minute with Yanni, showing that this was the agent within his scaffolds that really drove the production of cytokines by the cells that moved into it that aided healing. And so it allowed the scaffold to turn into a factory of healing in a way that wouldn't have happened if it was just collagen on its own. And we also developed frameworks to understand healing across a range of different donors and in response to different inflammatory transitions. But really what this fundamental insight allowed us to do is result in the product I showed in the slide before that can be taken off the shelf as a dry product and aids the new bone formation. One of the other things that I wanted to at least mention, because it's a sidelight that I wanted to share with Yanni, that has been coming up in the last couple of years, and we just finally finished the story this spring, and so I wanted to share where it was at. But within the wound environment, you don't just have the cells of the tissue you want, you have a lot of other cells that are there. And in the bone environment, we have the stem cells you might want to recruit, we have osteoblasts that produce new bone, you have osteoclasts that resorb bone, and you have immune cells such as macrophages that are all infiltrating in the wound environment. And as it turns out, it's very hard to study some of these processes in an animal model. My first project was an in vivo regeneration project, and after that I said, I really want to get out of an animal and actually be able to measure things for a while. Right? And so we started to imagine how could we create engineered tissue models outside of the laboratory to study signaling processes between these cells to make our scaffolds heal more effectively. We made two really major observations, the first of which is the stem cells that got recruited into our mineralized scaffold. We could optimize scaffold properties so they produce the factor known as osteoprotegerin. This is the factor that shuts down osteoclast activity. Right? So these are the cells that resorb bone. But right after injury, you want to be able to produce as much bone as possible. And so we were able to optimize the material, not only to have it produce bone, but able to shut down the cells that were degrading this new bone for a period of time so that bone could mature. It allowed us to more effectively get the implant working and optimize the material. We similarly used these types of tissue models to study how the process of immune response, thinking about, well, the stem cells and the immune cells that are moving into the scaffold are highly inflammatory. How can we tailor this material so that the inflammatory response might change? And this is the work that was here, that is the part I really wanted to talk to Yanni. When I started in Yanni's lab, he mentioned how early on they were designing these collagen scaffolds, and they went through a whole bunch of different chemical formulations, and the metric that they were always using to measure the quality of success was cell-mediated contraction, and was their skin at the end, was it skin or scar at the end, and how much contraction did they see? And there was a particular proteoglycan, a chondroitin 6 sulfate, was the scaffold that always led to the best healing. They tried lots of other gags and saw less quality of healing, but we had the long-term metric of scar or skin, but we didn't know why. And so we started a project where we started playing with the different proteoglycan contents using the same glycosaminoglycans that Yanni used years ago in our scaffolds to figure out how it affected healing. And we showed that we could take monocytes from patients, turn them into inflammatory M1 or healing M2 macrophages, and look at how the scaffold itself affected this kinetics. And what we identified is of all the different scaffold environments, the one that is this one right here is the dermal regeneration template. It allowed us to get the most rapid process to go from inflammatory cells to healing cells. So what they had been measuring years ago was cell contraction. The underlying mechanism was likely to do with activation of the macrophages that were moving into this wound environment. And so we've been able to do quite a bit of mechanistic work that, I think Yanni would have enjoyed, thinking about how do we make a better material that heals faster. We think about including mineral, including mechanics, playing with proteoglycan content that allowed us to show the kinetics of how immune cells interacted with these materials, and how the material that they optimized years ago is really the best in terms of how immune response behaves. So we have a material, it works great for bone repair. Let's talk about getting it to work for real. And so I had to put a nod out to Lawrence. You might recognize a few of these slides. We did all of our work in rat models and rabbit models. And one of the big draws for me to move out to the prairie in Illinois is that we have a massive College of Veterinary Medicine, the ability to do regenerative medicine's challenges at scale. And so working with our vet med surgeons at the U of I, we were able to create craniofacial bone defects in Yorkshire pigs. So these are 200-pound pigs. So we're creating defects that are now clinical scale as opposed to a tiny defect in an animal. These are large-scale defects, and we run into lots of challenges. The first of which is that try to get a surgeon to make a uniform defect in a pig, and you're going to have a mess, right? It's going to be a rough margins, and so you would have a material you try to implant, and it'd be bouncing around inside the implant. You get scarring around it. These pigs, the moment they wake up, they start chewing on the bars, and the material would get dislodged, and so the mechanical part of this was really challenging. And so we were faced with a biomaterial that worked exceptionally well for bone regeneration, but anyone who ever held one of Yanni's scaffolds knows it is incredibly soft. Right? It is not mechanically loaded, and any of these environments really are. And the mechanics behind how that works is what I spend a lot of time with Lauren, so I'm going to try to not screw this up. But if you look on the right here, this is a polyurethane open cell foam similar to what might be in the seat cushions that you're sitting on. And these foams are used in lots of applications in building materials, aerospace, and biological systems where you need efficiency. You need something that's strong but light, right? And the mechanics of how they behave is really interesting. As you start trying to squish it, the material will start bending. The individual fibers of these pores will start bending as the foam starts crushing. And you get this nice, if you have a stress strain curve, you get this linear zone where you load it a little bit, and it deforms a little bit, you load a little bit more, it deforms a little bit more, right? And at some point you get to the point where you've loaded it enough that the entire pore starts collapsing, and they all fall down on top of themselves. And you might see it if you had a honeycomb model like we used to make here, and you start loading it, you get a little bit of failure, then a strain band, and everything collapses on top of itself, right? And so the mechanics of this is that this elastic modulus scales as the relative density of the materials squared. And so all of these porous scaffolds are amazing for bioactivity because they're really porous. Cells can grow into them, they can get nutrients and oxygen, but mechanically they're very weak. You can make them stronger by adding more material, but then they would become incredibly dense and then cells would not live. And so we had to come up with a solution for how to adapt this. So this turns to some work of a past graduate student in my group, Harley, who got really fascinated with the idea of generative design, how we might create multi-material composites to solve a clinical applicability challenge. And so on the left is a example that some of you might have seen, or maybe you own one of the Futurecraft shoes that's made by Adidas, right? These are these porous soles that are produced by 3D printing. And the way it works is Futurecraft, the soles produced by Carbon 3D, have a series of porous networks that they have different cellular solids that are different densities and pore sizes. So they have softer and stiffer versions, and they created a generative approach to create a single sole that had stiff regions and soft regions, right? But this is a size 8 sole. You need to print a size 13 sole, right, or a size 12 sole, whatever you have. And the challenge we had for biomaterials was slightly different. We wanted a single material that could be cut and shaped on the operating room so that it could be used translationally. And so what Marley started thinking about was starting to produce Voronoi architectures. These are these random 3-dimensional foam structures. You start by a series of seed points where you connect them and it forms a 3D structure. You can make them denser or less dense by changing the number of seed points. And these materials can be printed now with 3D printing. And so this is a millimeter scale fiber architecture that has the same sort of collapse plateau and densification structures you see in normal cellular solids, except these mechanics can be at much higher scales. And so the solution that we came up with is you create these large mesh architectures that we embed into our collagen suspensions when we're freeze drying them. So this is us putting our meshes into our collagen suspensions, and then after they're freeze dried, they're turned into a single phase multi-material composite that can be cut or shaped. And so this is a single material. You can cut it with scissors or a razor blade, or interoperatively you can cut it with a cutting tool. But the interesting thing about this is the mechanics of these materials is driven not by the structure of the material, but by the relative density. So you can cut this in half, and it has the same mechanical properties as the whole. And so what that means is that we can now take a material and cut it slightly large, squeeze it down, put it into the defect, it fills space and springs back out. And so we can have a material that is infinitely adjustable, and it increases conformal fitting, and it allows better healing. And this has turned into a product that we're now talking to the FDA about, that is a collaboration with my colleague Justine Lee at UCLA. We have our mineralized collagen scaffold where we've embedded it with a PDLLA a graft that's modeled after the Resorbex plates that are used in a lot of craniofacial surgery. So this is a 3D printed PDLL plate that's embedded into our scaffold, and the entire thing is implanted in one phase. As we see over here, the defect heals a little bit. We put the plate on there. There's not really regenerative healing associated with the plate. Our scaffold gets good healing, but when you put them together, we get exceptional healing. It heals back faster, it heals back stronger, and it's mechanically protected. And so this is now the basis for a clinical product that we're in the final stages of of development and going to the FDA to actually get this into implant. And so it's been a long road, but it started with an observation of how do we make a mineralized scaffold for bone. It turned into mechanistic studies that allowed us to gain insight about how these materials work, and then it ended with a clinical solution that we're really excited about getting towards patients in the next couple of years. We're also turning our attention to the next mechanisms, as Yanni would want us to have done. And one of the next challenges we have is how do we improve healing at this implant defect margin? Again, I mentioned earlier, if you're older, if you have diabetes, if you have irradiated defects after injury, your implants may not work as well. Unfortunately, studies of implant defect integration are slow, they're expensive because they're in animals. Right? It takes a long time, you get very little insight. And so we've been thinking a lot about how to develop next generation tools to study this more effectively. And so we have a wound model system we've developed in our lab that is based on a hydrogel that is embedded with cells, vascular cells and stromal cells, that we then create a punch defect into and can implant our scaffold within the defect. This allowed us in high throughput manner to rapidly study cell infiltration and cell activity. And we've been able to replicate our data with large scale materials in the laboratory now in these wound models where cells are moving into the scaffold, they're going through the osteogenic differentiation process, and we can then expose them to clinically relevant conditions. We also can use not just the scaffold, but what's remaining in the margin. And we've been able to show that the cells that remain in this wound margin are also responsive to implant. They show inflammatory response short-term after injury. They show the ability to respond to the factors produced by the cells, and they can respond to other types of clinical inputs such as radiation in the context of healing after cancer resection. And so we've really developed a framework now where we can not only develop a material for clinical repair, but think about how we can optimize it for the next generation of patients. We have a lot of other areas in musculoskeletal repair that I didn't want to go into. I just wanted to mention one last one because I was back here and having a conversation with John this morning about where we're going with manufacturing. I have shown you materials that we've been making the way that Yanni has been making them for the last 60 years. We're making sponge materials, we're cutting them and shaping them, we're coming up with clever mesh composites that make them more clinically applicable, but we're still fundamentally making materials far in advance of how they're using them. And I think there's a lot of challenges going forward. How do we think about coordinated growth and how we think about bringing together ideas of manufacturing, sensing, and prediction in order to build better models of tissues, particularly to understand disease progression. And so we've been, the last number of years, working on a project related to the growth plate. The current tissue engineering model that I said was broken is you take a biomaterial, you dump a lot of cells in it, and you try to grow it back. What we think is a much more effective model going into the future is how growth plates operate. You and I were all born with much smaller bones. They've grown, and how does that work? We have fibrocartilage zones at the ends of our long bones that contain progenitor cells. And over about 15 years, they produce all the cells and all the matrix we need to grow a bone, right? So they don't have all the cells we need at the start. They grow them out over time, and they're coordinated, which is really fascinating. So how do you start thinking about the tissue engineering version of that? We've been starting to create these assembloids of stem cells and immune cells from patients that are in a living material, and then embedding them into our biomaterial to look at how different nodes interact with each other. Can we actually coordinate a process similar to what happens in growth as a way by which we might imagine a generative produce tissue engineering solutions as opposed to having a starting point that we have to modify? And we're really excited about the types of technologies you need to bring in synthetic biology methods, human tissue models. There's also a whole fraction about, what are the ethical, legal, and social implications of starting to create patient-specific tissue models to study disease progression, but we're really excited about the sort of manufacturing framework that goes into. All right, so that was my regenerative medicine. And in the last few slides, I want to talk about two other small things that I think are really exciting as well. As Yanni said when I was in his lab, spend a lot of time talking to people, get inspired by people around you, and think about what other directions you can move in. And so we started as a lab interested in producing biomaterials to implant for regeneration. And I started to have, as an assistant professor, lots of conversations around the U of I about, what does it take to make tissue models? These are now the rage, where people are starting to think, we're replacing animals with tissue models. What does that mean? Well, tissues are all hydrated, they're multicellular, and they're involving. And we wanted to think about how to make them. And so over the last 20 years, my lab has also engaged in the basis of building models of tissue, never to go back into a patient, but to study disease progression and therapy. And when you start thinking about how do you assemble a tissue, you first start with cells and maybe cytokines or biomolecules. You can add in a biomaterial matrix, and as a biomaterials lab, we spent a long time developing a whole toolbox of different gelatin-based systems. Gelatin being a denatured form of collagen. We have collagen scaffolds and gelatin hydrogels. And these are materials that we can cross-link together with lots of different mechanisms using light or enzymes. It allows us to create microdroplets or other types of systems. And lastly, we started to think about how do we develop materials to look specifically at multi-cell interactions. If we want to grow out tissues, we have to understand how cells interact directly and how they interact through soluble factors in remodeling. And these are fundamental aspects of both short-term changes in tissues and also long-term processes of healing. And so this became the basis for conversations at U of I, where a bunch of us realized that we were all thinking about applying tissue engineering approaches to understand cancer. And this is what has led, as you stole my punchline, right? But we're really proud of it that you're actually the first group who have been able to tell this in person, right? Is that about a decade ago, a few of us started going to lunch saying, we're all faculty at U of I working in different spaces where we're starting to think about cancer. We want to think about how engineering tools can be not just used for cancer, but developing new tools to think about how we treat and understand cancer progression in the future. And we set a goal to become a national hub for engineering and basic science applied to cancer. And the NCI designates basic and comprehensive care cancer centers. Comprehensive care, the metrics of success are clinical trials. Basic cancer centers, we have one here at MIT, one at Cold Spring Harbor, one at Jackson Labs, one at Scripps. There's 7 of them in the country. As of 2 days ago, there's 8. So it's the first new one since 1987. I don't know why I'm emotional about that, but we spent a lot of time in the last decade becoming the first new cancer center recognized in almost 40 years. And the first one that recognized that was explicitly from the perspective, how can engineers step into a hard challenge and build new tools? And as Myron mentioned, I lead the Cancer Engineering and Biological Systems Working Group. We're about 43 faculty members in this program. And our goal is to understand pathways and mechanisms that lead to cancer progression, develop new therapeutic targets in anti-cancer chemistry. So this is everything from small molecules to immunotherapies to whatever's coming next. Develop synthetic oncology models, so engineered tissue models and veterinary patient models in our vet school, and have translational impact. So this gets back to Yanni. Have a discovery, make it work, right? And so this has been a labor over the last decade to get to the point where we can be recognized. So I want to tell you a little story about one of the things that we do within this. We realized, as Myron said, glioblastoma is an incredibly terrible disease. I want to start with that. It is the most common and lethal form of primary brain cancer. Median survival is 15 months. That is up 3 months since 1999. And in fact, if you look back at historical rates, we have about a 5% 3-year survival. My lab, we do something called a journal club. Those of you graduate students know you have a journal club, you read new articles, you talk about them. We started a thing that was a throwback journal club where you had to bring a journal article that was older than you. That is getting easier as I get older, right? But that turned into a competition amongst my graduate students who could find the oldest journal article that related to their project. So we have articles from the 1920s in the British Medical Journal talking about what was glioblastoma known as then, had a 5% survival rate of 5 years. We're doing no better than we were in the 1920s. It turns out there's a lot of reasons for that. It also turns out glioblastoma is an amazing system in which you want to be developing engineered tools. How does GBM work? Well, it's typically diagnosed at late stage with some neurological deficit. You go through a debulking process where they try to cut out the tumor. And if we take a comparison to breast cancer, for example, you worry about breast cancer with metastasis. And so you think about taking out the tumor, taking out a margin, doing a full mastectomy. How do we get all the cancer out as possible? Obviously, you can't take the whole brain out. So at some point, you're putting a sharp surgical margin across cancer cells that are spreading diffusely in the brain. Right, and so this leads to rapid infiltration, rapid recurrence. About 7 months after you take out the tumor, you see tumor coming back almost always within like a centimeter of the margin. So what that means is the cells you can't cut out are the ones that are coming back to kill you. So this is a really elegant problem to try to think about. Here's our primary tumor environment. We got cancer cells, necrotic cells, a stem cell fraction, vasculature, parasites, astrocytes. We have macrophages and microglia. This is all cut out, right? That's the first thing that happens. What's left is the edges. And we got really excited about the vascular spaces at the edges of glioblastoma because there was a lot of literature suggesting the vasculature was a highway for glioblastoma cell migration. They're migrating along the vessels, not into the vessels. They're being exposed to a diversity of matrix proteins they never see in the brain except in the perivascular space. They're seeing cells they don't typically see, and they're seeing metabolic changes because as glioblastoma cells migrate along the vessels, they co-opt them, which leads to regression. So the vessel dies, the cells become highly necrotic and hypoxic, and then they hop to a new vessel. So they're seeing this diversity of matrix and cells and metabolic stance that are very hard to study in vivo, but you could imagine engineering systems in which we could create those. We also have colleagues in the Cancer Center that are specifically looking at GBM interactions with the immune system, such as microglia and macrophages, which are typically not found in the brain, but are found in cancers, and also the effects of radiation damage and large-scale delivery. But I want to tell you a quick story around how we started developing model systems to study vascular growth, and this is led by a previous PhD student in my group, Mine Yo, who's now a faculty member at the University of Wisconsin. And so she really wanted to adapt, Roger, the work that you have been doing, starting to create vascular models in fibrin gels, and she wanted to adapt them to our gelatin systems functionalized with hyaluronic acid to match the chemistry of the brain. And she started very simply using cell lines, a cell line of glioblastoma cells and cell lines of endothelial cells and stromal cells, and allowing the organization of these endothelial cell networks. You can see these red spindly shapes. Those are endothelial cell networks, and GBM cells are all green. And what she noticed in these early cultures was that it looked to her like the GBM cells were all getting closer and closer to the vessels over time, and that led to a series of observations. She showed that the process of putting these cells together led to co-localization and then regression of the vasculature. So they went through this regression and death that we see in vivo. They happen in the laboratory. She then developed microfluidic devices to show that the factors produced by these endothelial cells were able to recruit GBM cells closer and what they were. And then the real watershed moment for us is we were really interested in migration. And she came into my office and said, I've been messing around and talked to some people, and I think we need to look at drug response. And she had done an experiment where she started to put GBM cells and vascular cells together and was comparing the genomic signature of these cells to cultures that were separated. They never saw each other, but she just mixed the RNA after she isolated together to ask the question, how does them being together affect the way they behaved? And as we have the standard heat map, right? Lots of things up, lots of things down. The most highly upregulated gene associated with the co-localization of cancer cells and the vasculature was MGMT, a methyltransferase. And so the reason why we got excited about this is the frontline therapy since '99, if you show up with GBM, is you give you a drug called temozolomide, or TMZ. It is an alkylating agent that leads to cell death of dividing cells, but it turns out that if you have upregulated MGMT, you're able to remove this adduct on the DNA and survive. And so about 60% of patients that walk in the door with newly diagnosed GBM have high levels of MGMT and never respond to the drug at all. And the other 40% show production of MGMT over time. And her data suggested that the fact that the GBM cells that were close to the vessels would show really high levels of MGMT, suggesting they could be particularly resistant to drug. We tested that in a series of different models with our initial systems, but then scaled it out with more recent funding, now developing engineered vascular models with brain-specific cells, so brain microvascular endothelial cells, pericytes, and astrocytes. They form these really elegant vessel networks where you see perivascular wrapping of the pericytes around the endothelial cells. You see astrocytes forming end feet on these structures. They're laying down basement membrane. They're forming tight junctions. We're able to show that we can create a series of different levels of maturity of these vessels and start asking questions now using patient specimens from Mayo Clinic. And so we were able to now, in this case, embed a cancer organoid of GBM cells into these vascular networks and ask the question, how are they invading, how are they interacting, and how are they responding to drug? We're able to show that this vascular network drives more cell invasion. We're able to also show that the cells, the GBM cells, they start to migrate, they get closer and closer to the vessels. And so this data, what this is showing is from the light blue to the dark blue is increasing levels of maturity of these blood vessels. What we see is that as the vessels get more mature, the glioblastoma cells get closer, and they're less likely to be far from the vessels. And in fact, if you look at the stem cell fraction that is particularly bad, they get even closer to the vessels. So we see stem cell fractions that are highly organized around these vascular structures and showing high levels of drug resistance. And so this has become a model by which we're now testing a series of new compounds that were developed by our chemists in the Cancer Center as ways to get around drug resistance. It's also inspired us to think about new ways to create more adaptable systems, because this is one patient specimens that took 2 years to do and develop out. So we wanted to think about scalable approaches. And so this gets back to my other mechanics sort of aside. One of the things that I think has been lovely about the time at U of I is thinking about ways to develop new systems. There's been a lot of pushes over the last few years. You've probably seen some talks or read some papers about granular biomaterials. This idea that instead of having a big block of jello, we're going to have tiny microparticles of hydrogel we pack together into beds, right? And the interesting thing is there's been some fantastic work. Jason Burdick, Tatiana Segura, lots of other folks are really leading the field in producing these structures. What they're doing is they're producing empty space between hydrogel particles where the cells can live, which is exactly what Yanni has been doing since 1970, right? Is engineering porous space that cells would live inside of. And so we took this as a challenge to think about how can we adapt granular systems for cancer models. And so working with Andres Garcia's group down at Georgia Tech, we adapted some of our hydrogel chemistry to produce gelatin and hyaluronic acid modified gelatin versions of our hydrogel microdroplets. We produce a lot of them, but the different part is we're encapsulating our cancer cells inside of these droplets. So what does that mean? So it means we can take a cancer specimen out of surgery at Mayo and have them immediately into a hydrogel that matches the chemistry and mechanics of the brain and never sees tissue plastic. Right? It means we can now move these around, put them as packed beds, and actually create packed beds with multiple cell types. We have microglia and cancer cell versions. We can create mosaic tissue models to study progression. We can control the number of cells per droplet and be able to produce a highly organized structure. But that led to another mechanics problem, and I know I was going to come here, and I thought I was going to talk to Gareth, so I put 2 slides on here that I was terrified to show to Gareth, but then he's not here, so it's all great. So we got really interested if we're making microparticle, his loss, right? He can ask me later. We have microparticles. We want to start packing together to form beds. These microparticles are likely to not all be uniform, or if they start uniform, they're likely not to end as uniform. There's cells inside of them with producing matrix and degrading them. So we got really interested in how do these granular systems behave? You know, if you want to create more complex structures, you need to print them. So it means you got to flow these particles and then they have to seize up. So we started thinking about how do we describe the behavior of complex mixtures of microparticles? And we started thinking a lot about the idea of ductility versus brittility. And so this is work with Simon Rogers, who's an exceptional friend and a colleague of mine at Illinois, where we've been developing tools to understand how particle beds move into form. This idea being that we recently published, I think, a really wild paper that has a single yield stress fluid model to describe not just the flow conditions, the solid conditions and the flow conditions of a granular system, but how they change as a function of the heterogeneity of the bed. So this allows us to have a factor we call the brittility that is effectively the local versus global property of a gel that allows us to describe how this material deforms at a local scale as we load it. So it gives us new insight about how to move and print these microparticles filled with different cell types that are evolving in time. The first test of this has been some work now going back to glioblastoma, where we're embedding glioblastoma cells into these microparticles and then using with the robotic systems and high throughput drug screening facilities to physically pipette and move all of the particles and drugs in high throughput arrays, so we're no longer touching anything with hands, right? It's all going through an automated process where we can show dose-dependent drug response patient specimens in a high throughput manner. And so we're really excited about this is a new tool set because instead of us having to create these big blocks of hydrogel and let cells grow over weeks, and we can do this all really rapidly in an automated fashion. And in fact, we can, going back to our vascular networks, we can actually grow vasculature in the spaces between particles. So we actually now have a version that has high density of vascular networks grown in between the particles that are replicating this idea of drug resistance. And so this gives us a new leg up to think about how do we rapidly test new compounds for therapy. One last project I wanted to talk about, and it's weird to present in front of colleagues of your advisor. It's weird to present in front of your colleague that you're collaborating with as well. And so one of the other delights of being highly collaborative is that one of I think the longest collaboration I have is with Kate, so Kate Clancy in the back of the audience. So we've been married for a while, almost 20 years, right? But her expertise is in the biology of the endometrium. And so we, a number of years ago, got thinking about the endometrium is this in the lining of the uterus. It's a mucosal tissue that goes through huge amounts of dynamic remodeling across the menstrual cycle, right? Growth and resorption of vascular structures at a scale that any tissue engineer would be envious of. And so we started thinking about, are there ways that we can merge our interests in endometrial biology and tissue engineering to start thinking about new platform technologies to either understand physiology or pathology. And it started as projects understanding, could we create vascular models of the endometrium to look at trophoblast implantation, early stages of maternal-fetal interaction. Some of these early projects were tied to, can we create decidualized vasculature inside of a hydrogel model? And so this is the process of preparation that happens in the endometrial vessels before trophoblast invasion and the formation of the placenta. It also led to what I think is the first published work starting to put sex hormone binding globulins in biomaterials as a way to control the availability of sex steroid hormones. Most growth factors we work with are big and are easy to sequester and move around, but sex steroid hormones like estradiol and progesterone are incredibly small and don't. So we came up with ways to use sex hormone binding globulins in materials to control their availability. This led to fundamental questions, back to Yanni's drive, always find the fundamental question you want to dive on. And this came out of a question around how the vasculature of the endometrium organizes. It's often described as spiral arteries, these dense networks of spiral arteries that form, and after embryo implantation, you see this huge budding of spiral arteries to form the initial support for the trophoblast until the placenta forms at the end of the first trimester. But how does the spiral structure form, and why is that unique in the endometrium? And so this started a collaboration with Greg Underhill's lab at the University of Illinois, who develops high throughput two-dimensional substrates that allows us to look at the role of matrix properties on cell response. And we started asking a really fundamental question. Are the endothelial cells in the endometrium chiral? Is there a chirality associated to the cells? Is there an organization or a chirality associated with them that's unique to other endothelial cells? And is that chirality important? We were able to show in a really fun fundamental work that endometrial endothelial cells are incredibly chiral. Their chirality is dictated by the matrix environment in the endometrium, and that chirality directly leads to the degree of complexity of the vessels that they will form. And so it gave us new insight of how these vessel networks even form and resorb in the endometrium. This has led to the last project, which is our current project, that is the extension of forming the endometrium, is now looking at pathologies, and the big one has been endometriosis, particularly endometrioma, which is the lesions that form on the ovaries. So endometriosis is an incredibly common pathology. About 10% of all menstruators have some degree of diagnosed endometriosis. These are lesions in the peritoneal cavity, they're lesions on the ovary. They're incredibly difficult to diagnose. It can take up to a decade to diagnose because there's no good way to do it other than surgical approaches, right? And so, and there also, as it turns out, while there are mouse models to form lesions in the peritoneal cavity, there's no mouse models to form lesions on the ovaries. Is we have no insight of how these lesions actually form, because by the time we see them clinically, they're a decade old, right? And so this led to some questions. Could we understand the formation of lesions using this idea of matrix tropism? The matrix environment is shaping why lesions are forming here or there. And so this led to some work with Kate, Romana Novak, and Greg Underhill at the U of I, that we started to show that the matrix environment on the ovaries provides selective signals for both the epithelial cells and the stromal cells that are associated with endometriosis to attach. We developed initial two-dimensional systems showing how these cells collectively form adhesions. And more recently, we've now created our organoid systems using the same technology we use for glioblastoma invasion to understand how do these lesions, once they attach, why do they invade into the underlying stroma, and how do they become stable lesions? And in this case, understanding how epithelial cells tend to lead the invasion, followed by stromal cells into the underlying matrix. And more recently, looking at questions such as how does exposure to phthalates affect this process? We're all surrounded by microplastics and phthalates, and so there's a lot of questions about how our environmental factors may shape lesion progression. And so now we have tools that we use with cell lines, and more recently we're now starting to collect menstrual effluent samples from patients. So we get menstrual effluent samples that have all of the epithelial cells and endothelial cells and stromal cells, and we're using them to form lesions into the future. And this is a really, I think, exciting way to move forward, because it gives us insight into diseases using engineered systems that have been difficult to address. All right, so I'm getting to the end, and I have two last points I wanted to make. I realize we're all wanting to be done, but I want to just first thank you for the chance to talk. We talked a little bit about some of our tissue regeneration and cancer and complexity models, But actually, I think the tagline at the bottom that has evolved over about a decade in my lab really came from my time at Yanni. We want to do work that is collaborative, it's relevant, it's research-based, it's inclusive, it's responsive, it's innovative, and it lets us think about creating a new future. So I want to end with 2 slides. First of which, I work for a public research university, and it's an incredible privilege to be a professor, right? I think I've had a lot of conversations today with colleagues and students talking about what it's like to be a faculty member at this moment, and it's a scary time, right? This is a time where we could argue public trust in science, but I think more fundamentally, there's a privilege, a responsibility, a need for leadership to think about what it means to be a public research university. The thing that's really amazing about the Big Ten, where U of I is part of the Big Ten, we have 18 members of the Big Ten, please. We keep adding members of the Big Ten. We have 18 members, but the thing that's striking is we have 750,000 students that come through our door every year, right? That is education at scale. More than a quarter of all the books in North America are in Big Ten libraries, right? This is something that is a commitment to not just being the work we're doing and the teaching we're doing, but it gets back to the compact that we have with our society. We educate people to go out and do amazing things afterwards. Every year we have new people coming in our doors looking for that future. And it's our responsibility to think about how do we shape spaces that allow people to come in, allow people to grow, allow people to go out and do amazing things, and 20 years later come back and talk about what they're doing, right? And so this is the hard slide, right? We are at a moment of a lot of challenge, right? I am blessed to have gone to 2 amazing private institutions of higher education throughout my career, right? And we are at a moment where things are very uncertain, right? We can look around and say, there's lots of higher ed places, particularly private higher ed, elite private higher ed places, that have made decisions over the last year that I think are really complicated, that are really hard, and are making it so that it's hard to imagine what the future will look like. But I want to leave it this thought. Regardless of this moment, there will be an after, right? And the question is, at what cost will we have an after? And what are we doing to make sure we're committing things? And the pictures I have on here are probably self-explanatory. Probably recognize Columbia. Probably recognize Brown. This statement you might not recognize, but this is the thing that I was told I had to put in all my grants starting last summer, saying that even as public research university, I would have to put a statement in affirming that I would do nothing to go against any executive order that had been released by the president right now, whether it had to do with diversity, whether it had to do with gender ideology. If you think of, I've had grants frozen because we talked about menstrual effluent or menstruating people. Like, this is a moment of real precarity. And we then have the challenge that, we think about Epstein, right? And you think about particularly its connection with private universities and this private university, right? And what that means for us to think about, we need a future that people will engage in and believe in, and we want students to come here. We want students to think about what it's like to be in a place where we have smart, nerdy people that are trying to do the best for this institution and think about what the future should look like. This is a chance for us to really think about the value of public higher education, what it means to be a higher education faculty member in this university. And I'm really excited to think about the future that we can co-create together, because there are challenges, but there will be a future. And I'm very much looking forward to be a part of that with you. So with that, I would normally stop here, thank the current people in my lab, the past alumni, our collaborators, the grants to pay the bills. But I do want to end on this one because we do want to remember Yanni. As it turns out, I was talking to Ellen. I did not take nearly as many pictures in graduate school as I thought I took in graduate school. So I tried to find every picture I could, and this was it. And one of them isn't even from graduate school. But this is one of our lab lunches. We went to Legal Seafood. Like if we would go to lunch, he would go to Legal Seafood. In fact, I was telling Myron I didn't go back to Yanni's office after I graduated, not because I didn't want to, but because whenever I wanted to see him, he said, let's meet for lunch at Legal, right? So we went to Legal. This is Yanni at our wedding. So he showed up in his amazing cream suit and came in and wanted to talk to us before we got married. So this is, we're supposed to not, be with each other, but he's like, I need to talk to the two of you. And so this is in Kate's living room. He wanted to come in and talk to us and, and share his wisdom, right? And then the last one was at the Museum of Science when they had the unveiling of the Hoper-generated video a decade ago when Yanni was inducted to the National Hall of Fame, Inventors Hall of Fame. And if you have not seen the video, it's 15 minutes long. It is transformative. It will give you hope in the future. And I think that's what Yanni gave. He gave me a chance to think about working on big problems making investment in people, and thinking about what are the next challenges that we want to build the foundation for. Because that's, in the end, what it means to be a faculty member at a university is you're not solving the problems today, you're solving the problems that your students will solve in the future, right? You're building the foundation where they can look further and do better, and that's something Yanni did his whole career. I want to thank you for the chance to come back. It's been really emotional to think about. It's been fun to show off some of the science for him, right? And I'm happy to answer your questions if there's time. So thank you. I hear there's also drinks outside.
Really, really great talk. Thanks for that. Thanks for the words at the end. All of them. I'm curious if you go back to, you mentioned, you talked about in vitro models.
Yeah.
And there's been this vision of, we're going to get rid of animals.
Yeah.
Hasn't really happened. I'm curious, in your area, what do you feel, are there types of problems you feel are better suited for in vitro models compared to others? Because at first it was going to replace everything, and now we maybe have come back more to reality. I'm curious, do you think there are types of problems that are better suited for that?
I think certainly. I think that, this is the question. I think this is something that a lot of us have been trying to think about. Like, my lab technically has been working on new approach methodologies since we opened our doors in 2008 and wanted to think about models of the bone marrow to study. And we had to convince the hematopoiesis community that what we weren't doing anything in mouse, but we could actually learn something out of it, right? And I think there are the idea that tomorrow the FDA may very well likely just do this, but what is the actual value we get out of tissue models is so far into the future that there are some limited cases where I think you have ways in which you can use these models to add insight. And so one of the thoughts that I think has been important is that in vivo models give you lots of complex data. You have an animal, you put an implant in, you got long-term healing, and you can only look like a couple of times. And the reality is for most animals, you only look once, right? Like, you end up building a time scale. Well, this was healing at 3 months, this was healing at 9 months, this was healing at a year. You get these very specific time slots that you can look at. And then you try to make guesses of how they connect. Like, how you go from this spot to this spot in time is really complicated. Like, we have our biomaterials we put into an animal model. A year later, this one worked the best. It's not necessarily because this one was the right one at the start. It means the evolution process or the remodeling process got to the point where it was at. So the thing I think that these new approach methodologies can use is try to start filling in the gaps between known in vivo data points where we have some assessment of differences, and we try to use the model systems to study there. So we've done it in bone healing. Has actually been a great one. We know quite a bit about the kinetics of bone repair. There's some inflammatory conditions we can use. It's a fairly well-defined defect. It's allowed us to gain a lot of insight with our wound models about how rapidly cells infiltrate and how that shapes healing. I don't think that scales to everything yet. And I would, if I was being honest, I'd say, I don't know. Like, we were talking this morning about, like, what is the manufacturing base you need to create to actually produce scalable systems that if we talk about assembloids or organoids and living biomaterials, how do we build small scale models that allow us to make a measurement? How do we bring in sensing tools? How do we bring in the ethical, legal, and social implications perspective as well? Because the moment you start doing these, you worry about, what's the biosecurity? We don't want to end up with the next Henrietta Lacks situation where we're doing some of this with engineered tissue models. We want to think about how to keep people's data safe or genomic data safe. We want to think about what does that mean in terms of variability. Every time we take a patient sample, we typically don't know how to deal with the variability we see. So I think I would love to say we'll get there in the next decade. I don't think so. I think we're going to build the frameworks to understand what a modular engineered tissue looks like. And we'll be able to use that to start to understand what types of transitions we can measure and what types of healing processes we can look at. I think acute injury is the first one because there's a rapid response. I think something like aging is a much harder problem because that happens over decades. And so I think sort of the acute injuries or, some sort of, cancer has been another really rapid one because of the speed by which you see progression. Those, when you have shorter times, I think we get more insight out of them. But as it being something that I think we're going to use tomorrow, no. But I think those are the areas where we'll see it first. But it's going to be a while to get to the point where we'll be able to trust it. But you know, as my friend Ed Lou, who's one of our advisory members on the Cancer Center, he's just retired as the CEO of Jackson Labs. He makes all the mice we all buy. He says, you know, the reality is that, like, you know, mice are just artificial realities anyways. We just know how to turn them into clinical insight. And so I think getting back to Yanni's, you know, vision in the beginning, the more we can use these new approach methodologies to identify the feedback to some sort of specific clinical insight, even if it's small scale, that will give us some benefit to use. But it's going to take a while to build out that infrastructure. Art.
From the beginning, you had me drawing on pore size and porosity, and then started looking very random. Then at the end, you said, I'm going to go homogeneous pore size. And one of the statements I would make is, you had a dodecahedron from one of your early slides.
Tetrachydecahedron, so I got to get that right, Lauren. Okay. Tetrachydecahedron, but yes, it's close. 12.
One of my projects at Granular was trying to make that. 20 years later, but I didn't take that project.
That was the project that Lauren and Yanni convinced me to do right after my master's, was how do you make really uniform pores? And that's why I spent all this time talking to Mert Flemings. So we ripped off what steel metallurgists did to get uniform grain sizes, to try to come up with thermal processing approaches to get really consistent ice crystal growth. Once we got consistent, that led us to align, so we get this directional solidification. And then that allowed us to make this multi-phase material. So we had to make uniform before we could make complicated. And so that was the workflow that got there.
The question was, what's the optimal pore size and shape for bone generation?
Well, so what I will tell you for us, it's about 150 microns in pore size, and it's anisotropic. It's an aligned pore, so we get ellipsoidal pores stacked together. It's about a 1.4 to 1 ratio, so you get this kind of minor ellipsoid. That those structures, you see really efficient cell infiltration. We also see really efficient activation of M1 phenotypes to kick off the inflammatory response of macrophages. But once it kicks off, they very rapidly turn into M2 macrophages that are pro-healing. And so we went through a lot of the mechanistic stuff, was to figure out the right pore structure, the right mechanics, and the right rate at which the mineral components are eluted out of the scaffold. So we have no osteogenic factors in there, except for the fact that we have calcium and phosphate that are being released slowly out of these brushite crystals we form. And that leads to a really efficient osteogenic effect that we don't need to add anything else to, which from a surgical standpoint means this material is sitting dry on the shelf in the operating room until they cut it, shape it, put it in, and it's done, right? And you just walk away. And so from a translational standpoint, that is really nice. We're looking at versions that are more complicated for, you know, diabetic defects, for radiated defects. These complicated defects, we have a next generation product line that has more stuff added in that's harder to do, would be harder regulatory wise to get approved, but would be important to boost healing. But the native one is it is the dermal regeneration template with a tweak in the structure and incorporating a clever way to get mineral in and out of it. And it works incredibly effectively in bone. So it took Yanni's skin to nerve to bone. So it still works. Thanks. Go.
Yanni, President, my wife died of glioblastoma 6 years ago. And be a faculty member, one of your former students, making headway on the disease that killed my wife. Brendan, thank you. Thank you. Thank you.
Yeah.
We should wrap it up, but just wanted to say thanks so much, Brendan. That was a wonderful lecture. Thank you all for being here. Brendan's family are here. Thanks for coming. And Yanni's family are all here as well. So it's really special to have you here to honor your father and your mother. Lisa told me to tell you all these drinks and food outside, so please enjoy. But thank you, Brendan. That was really amazing.
Thank you all.